Well, welcome everybody to Optometric Education Consultants National Webinar Series, Summer's Tuesday night edition. Our speaker today is Dr. Nora Cothran, who is commissioned as an intelligence officer in the United States Air Force. She was selected to serve as the Director of Intelligence for America's first combat-capable F-22 Raptor Squadron and actively contributed to national security as a contracted intelligence analyst for the Department of Defense. She's a Southern College Optometry graduate, and she did a residency in ocular disease at Bascom Palmer Eye Institute in Miami. She's a fellow of the American Academy of Optometry, an active member of the American Optometric Association. She serves on the AOA's Coding Reimbursement Committee and the Pinellas Optometric Association's Board of Trustees. In 2023, she's awarded the AOA's President's Award for contributions to the profession. She lives in St. Petersburg, where she specialized in the medical management of glaucoma and neurologic eye diseases at the Eye Institute of West Florida. So, Nora, we're really happy to have you. We're thrilled. It's our pleasure and privilege, and the audience's pleasure and privilege to hear you talk about glaucoma. So, Nora, please take it away.
Awesome. Thank you so much for having me. It's kind of fun to, I remember, watching y'all's lectures and seeing you in the lecture space, and to now actually be here as, a guest on your platform is a true privilege. So thank you very much. Tonight, we're going to talk about my favorite topic, which is glaucoma. And the title of tonight's presentation is, Are You Ready for It? I am a self-declared Swiftie. And so are you ready for it? Because we're going to get up to date in the glaucoma space, technological innovations and a shifting paradigm. These are my financial disclosures. This slide deck was planned and prepared solely by me without any input from members of any of the ineligible companies. And these are our course objectives. The goal of tonight's course isn't to, make you a glaucoma expert, but I want all the doctors that are on the call tonight to walk away feeling just a little more confident in what is out there in the glaucoma space here and now. An overview of what we will discuss. And this is a 2-hour course that will be obviously a little less than 2 hours, but we're going to be breaking it apart into little chunks. And then throughout each of these little chapters at the end, there will be a polling question or two. And y'all are ready for that to look in the top right-hand corner of your platform where Greg showed you before. So let's talk a little bit about background. What is glaucoma? That is literally the $1 million question. I get asked this all the time. What we need to understand is that glaucoma is actually a family of optic neuropathies. It's not only the leading cause of irreversible blindness in the world, but in the United States and worldwide, there's a multifactorial pathogenesis. So there's an anatomical component, a vascular component, and in my humble opinion, I think there's probably an autoimmune component that's out there. Ultimately, at its root, it's a neurodegeneration of cranial nerve tube. This is a disease that can be aggressive, and in later stages, it can be recalcitrant, meaning that it is not easily leaned toward therapy. It's estimated that approximately 4.2 million Americans are currently living with glaucoma, and over half of them don't know that they have the disease. Within the optometric space, we're rising to the occasion. I see my colleagues out in all the different facets of our profession learning a little bit more about the disease, even if they don't actively manage and treat it. They're looking for the early indicators of disease, finding patients that have family histories, because we know there is a genetic component to this disease. 128 loci have been identified, and there was another paper that showed, our understanding of the genetics of this disease is just blossomed in the last 5 years. This is a disease that disproportionately impacts the African-American members of our community, and patients that have low health literacy, those that are unemployed, disabled, and those that speak English as a second language have much poorer outcomes from this disease. This is a disease that has not only a physical factor, but there's a psychological component to this disease, both in the management of the disease as the doctor, but for a patient that's suffering from this disease. When surveyed about a hypothetical loss of a sensory organ, patients are most fearful of losing their sense of sight. It's not losing their limbs. It's not losing their sense of smell or touch. It's actually losing the sense of sight. And just in the last few years, as physicians, I feel like we're really understanding that there is this intangible component of vision loss that we are starting to understand, that we're realizing that the way that we communicate with patients, the way that that we discuss therapies, there's an art and a science to managing this disease. And when we have so many new tools in the toolbox, we're now realizing that we can still focus on the foundation of the science that we have, but we actually really get to practice the art form of managing a progressive blinding disease. What is very sad is this is a statistic from the public health literature. Patients with severe vision loss, not just from glaucoma, but other diseases like macular degeneration, diabetic retinopathy, these patients not only suffer from higher rates of anxiety and depression, but they suffer from higher rates of all-cause mortality, meaning their lives are shorter. So here we are as optometric physicians practicing in a space with this growing our growing scope expansion within various states, and we have the ability to actively manage a disease that could not only potentially have a patient enhance their life through maintaining their vision, but also could ensure that they have the longest, healthiest life possible. When we look at the pathophysiology of glaucoma, we really need to understand that the primary source of dysfunction of this disease is within the TM, the trabecular meshwork. I remember being in school and learning about the different anatomy in the anterior segment and the posterior segment, and the glaucoma section was solely focused on optic nerve, optic nerve, optic nerve. But we need to see where the dysfunction is that happens that ultimately results in the degeneration of that optic nerve. And it's at the front of the eye and the angle at the TM. We can get extracellular matrix accumulation that leads to then inflammation. There's ultimate fibrosis and sclerosis. Different types of glaucoma affect the TM in different ways. And a lot of the new research that we are understanding from the world of genetics and the sect of glaucoma genetics is really showing that there is a genetic propensity for dysfunction within the TM in and of itself. The chronicity of glaucoma leads to the irreversible dysfunction. There are drugs out there that work in the TM, but if you save them for later, you can't put a medication on board that works in an area that has already become damaged due to oxidative stress and due to long-term inflammation. And so ultimately understanding that the trabecular meshwork is the source of the dysfunction in the disease is super important to remember. Remember, not all glaucoma patients have higher than average pressure, but the baseline intraocular pressure of every single glaucoma patient that you care for is too high for that particular patient's eye. 20, 30 years ago, it used to be thought that glaucoma was this high eye pressure disease, that everyone that had high eye pressure had glaucoma, and, average pressure, normotensive, low tensive glaucoma was fairly rare. And now that we're understanding to look out for these early notching, these Strangehemes, and trying to understand patients that have family connections to the disease, I think that we are really understanding that we have a disease that ultimately damages cranial nerve too, but across the spectrum of high pressure, low pressure, understanding the TM is where the dysfunction is, but knowing that wherever that starting point is for that patient, whether that number's 35 or whether that number is 13, that number's too high for that particular patient. Let's talk a little bit about diagnostics. One of the polling questions was actually which tool that you like to reach for early diagnosis of, glaucoma, and one of, the options was a visual field, or perimetry. And this is an area that I've really, I have enjoyed watching the space explode. I was an intelligence analyst for the DoD before I became an optometrist. And so I really love seeing new technology, and especially when that new technology can help people, because ultimately all of us took that optometric oath to care for our communities. The virtual perimetry space has exploded. I watched the first kind of rumblings of the possibility of a virtual perimeter, 8 to 9 years ago, watched them come to fruition. I've watched some come off the market. I've watched some rise to the top. And the Humphrey Field Analyzer, this was the gold standard for glaucoma field testing when I trained at Bascom Palmer 10 years ago. And as I moved into clinical practice, I saw that, that was the gold standard. But these type of static perimeters, they have a lot of limitations. They're very expensive. They have a very large footprint. Almost every office has one room that's dedicated to put this Humphrey field analyzer. And then also the cost of maintenance. The accessibility to me was the first thing when I first started, as a second year student at SCO. We did a lot of the testing to learn how a lot of these diagnostic tools work. And I remember seeing a patient that was in a wheelchair that was not able to get into the virtual or get into the static perimeter. And I remember thinking, wow, here's a patient that has one whole facet of, of technology that they're completely cut off from. And that doctor who's managing that patient's disease isn't going to be able to utilize that particular tool. And at that time, there was no such thing as a virtual perimeter. And I thought, wow, how sad that the patient now, their glaucoma, the doctor has to work harder to manage the disease, but the patient doesn't get the benefit of something like that static perimeter. So seeing these virtual perimeters come on the market would just warm my heart, because I thought back to when I was that student, seeing that patient that couldn't fit into the static perimeter. And so here's one of my patients that I've been caring for her using objective measures. This lady, she can't, she sits so low in her chair, she's not able to actually get into even an OCT. She definitely can't get into a static perimeter. And we now have a virtual perimeter that we were able to put on her. And for the first time in 9 years, we were actually able to, I was able to show her what she was experiencing. And what it does is it helps me to quantitatively measure not only what she can see, but also what she can't see, so that I can make sure that that area stays static in time. The ultimate question that I get asked all the time is, are all of these the same? They're all out there. A lot of them look similar. Some of them look different, but are they all the same? And I started evaluating the space as a former analyst and kind of seeing where where these perimeters differ and where they were very similar. A lot of the perimeters seem to use a commercial off-the-shelf system, and I saw that some perimeter companies were advertising we have X number of published data. So I wanted to always see the data. When I was an Intel analyst, my troops would come to me and say, "Oh, we found XYZ," but a lot at that time it was terrorist cells, but or interesting intel, and I would say, "Show me the data, show me the data, show me the data." And so when I went to look to see what some of the data was for some of these virtual companies, over half of the data, or one company like 90% of the data, was all for blepharoplasty patients. So it's great to say that you've got this nice device and that you have X number of published papers, but if the papers aren't really completely accurately reflective of the market that you're trying to reach into, I think that there's limited value in that. TVST is the journal of ARVO, and that's the Association of Researchers in Vision and Ophthalmology. And this is a paper published by Chris Bradley and his team last year, and it validated the algorithm. So it's not just looking at the hardware; it's actually looking at the software that runs one of these virtual perimeters. The Humphrey field analyzer is using something called the CETA standard. So when you hear CETA or you see CETA on that printout that you get for your Humphrey, that's an algorithm, the Swedish algorithm that is proprietary to Humphrey. The Radius platform uses one that's called the RADA standard. And this was a head-to-head comparison to look to see with regard to if this was not only repeatable, but also if the staging, glaucoma staging, mild, moderate, severe, is really important. This is important for billing and coding. This is important for knowing when we need to, say, pull the trigger for more aggressive therapy. And Chris and his team had 100 patients. They were in two kind of buckets of suspect to mild, or even moderate and severe. And the patients were randomized, either Humphrey radius, or radius Humphrey. And what they found was that the RADA standard algorithm was comparable to the CETA standard, and it was really high concordance with glaucoma staging. And so this is the first time that one of the virtual perimeters has published in a journal like the ARVO journal to show that it is on par when it comes to the software component of that proprietary proprietary algorithm with the Humphrey Field Analyzer, which to this point has been the gold standard. Why that's important is that allows optometrists to make very thoughtful decisions. Say their Humphrey dies, and they may want to have a ability to test visual fields without having to use all of that space. They can then make a choice for a virtual perimeter that's going to allow them to accurately care for their patients, but with a system that they know is going to be compatible with the disease staging. This is another tool I get asked about a lot, multifocal ERG. ERG is an electroretinogram, and what it is doing is it's looking at the retina, and the multifocal part means multiple ERGs across the retina. It gives a lot of good diagnostic information for a lot of inherited and acquired retinal disorders. That's really important because it's often used to monitor patients that have disease processes that are retinal in nature, or to evaluate for toxicity of drugs that impact a retina. Note to this point, I have not used the word optic nerve. I feel that there's limited clinical utility in the diagnosis and management of glaucoma. It is a hill that I will die on. I politely butt heads with some other members of other optometrists that say, oh no, this is really important for glaucoma management. So at the American Glaucoma Society meeting that I attend every year, I started just polling some of the fellowship trained glaucoma surgeons that were out there and asking them, hey, how often do you employ this in clinical practice? I stopped asking after I asked 29 of my friends and colleagues, because they all kind of laughed at me and they said, we don't use that. I said, that's a retina tool. I sit on the AOA's Coding and Reimbursement Committee. And the CPT 92274 is a code for the multifocal ERG. It's not reimbursable for any of the glaucoma ICD-10 codes, and there's a reason for it. So I always want, as an analyst in the military, and even as an optometrist, as a doctor, I think it's important for us, if sales reps tell you one thing, you need to use your own, best judgment and decision making. Because I will tell you that CMS is looking at our profession, and not in a good way. Optometry's utilization of this code in areas that are like optic nerve atrophy, and billing for codes that optometrists say, well, it's the same as glaucoma. It truly isn't. And there's a reason why you can't use the ICD-10 codes for glaucoma. So just because a rep tells you that something is okay to do, doesn't mean that you should do it. And I want you to think, will this tool, using this and this data, change the way that I'm going to manage the patient? And if the answer is no, think twice about, utilizing that tool. Because right now we are under the scrutiny of CMS, because our utilization of this code has exploded, and it is not on par with, say, fellowship trained glaucoma surgeons who manage glaucoma day in and day out. So I just want you to be very careful, because as the utilization of this type of a tool rises, potential revenue of the RVUs goes down, and it opens doctors up to audits, and puts us even more under the microscope in realms where we're trying to work for scope expansion. That's not the kind of attention that we really want to draw. So just think, is this going to really change the way that I manage the disease? And you can make your judgments from there. But those are two of kind of the hot tools that were out there diagnostically that my colleagues always ask me about. So let's talk about
Nora, for clarification, just for clarification with that device, it's good for the diabetic retinopathies. It's good for the retina, right? So I just don't want to there's a lot of optometrists that have this unit. They like it for the retina, but just be caution with the glaucoma side, right?
Yes, just be caution with the glaucoma, because remember, it's an electroretinogram. So this is utilized even, within, retinal practices, because it is a tool for the retina. And there are so many wonderful papers that are published to show that it can actually detect diabetic retinopathy early, before we actually start seeing some of the damages that manifest in the retina. So thank you so much, Greg. Yes, great for the retina, cautious with use with anything within the glaucoma realm. Let's talk a little bit about treatment. I have created this therapeutic timeline that starts in 1954 and moves on into the present day of all of the major classes of glaucoma drugs as they came onto the market. What I want the audience to look at is the distance between 1996, which I still probably had a Hello Kitty lunchbox, and 2017, when I was in clinical practice, started already in clinical practice, and we went almost over 20 years without a new drug class in the glaucoma space within our toolbox. In 2017, we had the rho kinase inhibitors and the nitric oxide donating prostaglandins that came onto the market. But within the last, 50 years are when all classes of drugs came onto the market that we utilize here and now. Prior to this time period, we had drugs like pilocarpine, oral CAIs that were, heavily utilized. But what we usually use in our toolbox now are yellow caps, purple caps, orange caps, teal caps, and our white caps are now just within the last 50 years. And so, my hat's off to the optometrists that we are, that paved the way for us to be able to be practicing the way that we do. I want to take a brief moment to discuss how FDA clinical trials are executed, just to refresh everyone in the audience. There are 3 phases to an FDA clinical trial. Phase 1 is mostly focused on human safety. Up to this point, a company may have had animal models or other in vivo, in vitro types of testing to prove a mechanism of action. But the phase 1 is mostly to focus on the safety in humans, whether it's if it's a pediatric drug within that patient population or an adult drug within an adult population. Phase 2 is looking at that medication concentration. So it's that molecule has been shown to be safe in humans. And we're looking at the concentration of the medication. And we're also looking at the dosage. So some companies may have 2 different dosages that they're looking at. Others may have 3, but we're looking at the concentration, the dosage, and also, and if we're doing it once a day, twice a day, 3 times a day. Phase 3 is what we usually hear about, the phase 3 clinical trials, and that's the investigational portion. The phase 3 is going to have a primary and secondary endpoints. These are endpoints that a company has to say ahead of time what they're investigating, and they are either going to meet or not meet those endpoints. Once the ball starts rolling for a clinical trial, you cannot change your primary, secondary endpoints. You have to keep moving forward with it. And most companies will be doing 2 phase 2, 3 trials that are identical to make sure that the information that they're garnering or the data they're garnering is repeatable. This is a chart that I created when I was studying for my boards a decade ago. I couldn't find any charts that actually showed the glaucoma medicines that were out there. So I just made one, and I am a visual learner. So I made this with the cap colors in the boxes of these drugs. And right now, this is a chart that shows all the available drugs that are on the market for the therapeutic management of glaucoma based by drug class. And then the intersection of the drug classes, you'll see the combination or the fixed dose combination drugs. Anything that has an asterisk next to it are those that are preservative-free free drugs that are out there. So we're going to focus on 2 categories of therapeutics. One category is the recently approved, and the others are the investigational. The recently approved are important because this is what you have today, tomorrow in your therapeutic toolbox to be able to reach for. Well, actually not. One of them is still yet to be determined if it's going to, how it ends up in our toolbox. It's been FDA approved, but so I'll step back one step. One of those we don't have in our toolbox yet. The investigational molecules, I think it's important for you to know kind of what's out there so that also you get an understanding of what's available in the space. Because I get asked all the time by patients. I just saw a patient, she's 19 years old. She's almost completely cupped out. She was diagnosed by her optometrist in a commercial setting. She went for some glasses. She just started college and was having really trouble focusing, was realizing that her contrast sensitivity at night wasn't the same as some of her friends when they were out. And she had undiagnosed glaucoma. And today it was my first time meeting her, and she asked me, she said, what do you think, is going to happen, in my life? And I said, I think that you're going to continue on with college, and you're looking through a little drinking straw area of space in each eye. You've still got that 20/30 vision in each eye. You have very little rim tissue left, but there are molecules out there that are actively in clinical trials that in our lifetime together are going to be there for you. And so my goal is to keep your scaffolding safe so that I've got something to build upon when these molecules come out. Because hope is super, super important in patients that are oftentimes feeling hopeless. So the first medication we're going to talk about, the first two were FDA approved in 2017. The first is latanoprostene bunod. It goes by the trade name Vazolta, and it works to increase aqueous outflow by utilizing the traditional and the non-traditional pathway. So the uveal scleral outflow and the trabecular meshwork. This is a prostaglandin analog, latanoprost, but it donates nitric oxide. So for the longest time, pharmaceutical companies were trying to get nitric oxide as its own molecule and thought, wow, what a great idea if it could be in a drop, but it doesn't stay stable on its own in a bottle. This is a molecule that's an endogenous molecule found in every human organ system in the body. And what it does within a vascular system, and it kind of relaxes blood vessels and smooth muscle cells. Nitric oxide producing cells are found everywhere, including the eye, and especially in the aqueous outflow pathway. And studies have shown that patients have glaucoma, they're 40% deficient in nitric oxide metabolites. It doesn't mean that the receptors aren't there. It means that those nitric oxide metabolites aren't there. So multiple companies were looking to try to figure out, how do we put nitric oxide into this space? And they were able to do it by combining the molecule, binding it into latanoprost. So when the molecule goes in the eye, it splits into two separate molecules. In the FDA clinical trial that brought latanoprostene bunod approval, it was a trial that was head-to-head with timolol, because at the time that's what the FDA required. These next two slides are an example of, if you as a doctor see something in clinical practice, then you have the ability to act on it to be able to get more information. I was an analyst, and I was noticing that when I would switch patients from prior prostaglandins, regardless of any teal cap that they were on, and I would switch them to this molecule, I was curious if all patients have that nitric oxide, the glaucoma patients have receptors for nitric oxide, and if they would respond to the drug and how much. Because head-to-head to timolol doesn't really tell you much about what the nitric oxide is doing. And up to this point, there really weren't any papers that were publishing it. So Dr. Connie Okeke et al., and I got to be part of the et al., and I was the second author on this publication in clinical ophthalmology. We did a retrospective chart review on our two separate practices, and we found that when we don't wash out patients in real life. So if a person has glaucoma and they're worsening, you don't just tell them stop all your medicines for 6 weeks and then put them on a different drop, like a clinical trial. We actually just switched them the same day. So we wanted to see patients that were switched from a prior prostaglandin, how much of a drop that they got. Our inclusion data was for patient that had 2 or more follow-ups. So the disclaimer is this does not include patients that are non-responders, because to me, clinically, I find about half of patients respond, half don't. I don't know why the ones that don't respond, but they just don't get a significant pressure lowering different to any other prostaglandin. But the ones that did got about 25% additional pressure lowering, which could be potentially attributed to the nitric oxide. 49 patients isn't a lot. So what happens if a larger pool of patients was evaluated? There is a registry called the IRIS registry, and that is a registry, largest registry in the United States. And we looked starting out with 4 million patients and pared 833 down to meet our inclusion criteria. And these are patients that were actually already being treated for glaucoma. And they had a mean IOP of 19.5 millimeters of mercury. And these patients needed to be changed to something different because their disease was unstable. We had 2 buckets of patients, those that were on prior PGAs, any of the teal top drops, and those that were on non-PGAs, so on fixed dose combinations. And those patients from the IRIS registry were switched to LBN. Their disease was progressing, whether on fields or they're 24-2 or they were above target. And they were switched to LBN, and we wanted to see what happened to those patients. And when we looked at the data to see those that were switched from LBN to LBN from a prior PGA, the ones that were on any prior PGA got about 2.4 millimeters of additional IOP lowering than what they were getting before. And those that were on non-PGAs, interestingly, so these would be all patients on combination aqueous suppressants. When they were switched and they were stopped that drop, put them on LBN, they were 3.3 millimeters of mercury lower than what they were before. The first I attribute to the nitric oxide, that first category on the left, and the right, I actually attribute that to probably some component of the PGA and the nitric oxide, but also something that has to do with compliance. Because if patients were on non-PGAs, they were probably on a drop 2 to 3 times a day. So this was a poster that I just recently presented a few weeks ago at AOA's optometry meeting, and it will probably get published. But this shows that even, as a doctor sitting in a clinical practice, what you observe with your patients, you can take that information, seek out more information and elevate it up. And it really helps us all as a profession in optometry and ophthalmology to understand what we have in our toolbox, how powerful some of our tools were. Speaking of powerful tools, the next medication we're going to talk about is Notarsadil. Notarsadil is a beautiful molecule. It is wonderfully unique, and it was the first new drug class in 20 years, the Rho kinase inhibitors. You can remember Rhopressa, Rho kinase. They both start with R's. And Rocklatan also starts with an R. Rocklatan is the fixed dose combination as Rhopressa paired with latanoprost. This actively works in the trabecular meshwork by relaxing, binding in, and blocking Rho kinase so that the TM fibers can relax. Some of the common side effects that you would know or be able to see from this drug are conjunctival hyperemia and petechiae. And that's what I noticed when I first started using this in my patient. Corneal verticillata, you can see in the picture here on the right, these little swirls that you see here, it will look like the amiodarone swirls that you see. And thankfully, they are not visually significant, and they may move from visit to visit when you see the patient. So don't be alarmed if you see this. This is not a reason. I remember when I first started using this, my cornea specialist was like, what is going on all of these corneas? And when I looked in the microscope, I said, oh, that's just verticillata. And he goes, well, do they need to stop it? And I said, well, no, if a patient has amiodarone swirls, do you have them stop their amiodarone? He goes, no. And I said, well, these were not shown to be visually significant in the phase clinical trials for Notarsadil. The one component of the cornea side effects is something called reticular bulbous epithelial edema. We call it corneal honeycombing. It has a very unique appearance, and it'll almost look like little bolus blistering. It oftentimes happens at the limbal margin. I've only seen a handful of these, and a few of the patients that I've seen them in, they have concurrent autoimmune conditions. So if you do see something that is unusual, you can always do a trial off the medication to see if this resolves. Rho kinase inhibitors, interestingly enough, this whole class of drugs, they're being investigated for use in treatments of corneal diseases like Fuchs endothelial dystrophy. So even though some of the side effects for this new drug class you might see on the cornea or on the conjunctiva, they actually may show promise and utility for treating diseases of the cornea. So this is a little picture that may show a little bit of some of the petechiae. It's a little bit hard to see. For some reason, my little phone, when I was taking this in the biomicroscope, was catching this reflection here. But you can see these teeny tiny little petechiae here. You'll see these sometimes as perilimbal flush. This hyperemia across the conj. It's very different than the hyperemia that our colleagues may have seen when the prostaglandins first came to market, and it's definitely different than the hyperemia that you see from a bromonidine hypersensitivity. This is not an allergy. This is a known side effect, and it is usually purely cosmetic. I talk to my patients and I explain to them ahead of time, it doesn't burn, it doesn't hurt, it doesn't sting, but your eye might look a little bit flushed, like you got splashed with swimming pool water, and it usually fades in 4 to 6 weeks. And truly that's what happens. So if you are prescribing this medication to a patient, don't hesitate to tell them what the side effect's going to be beforehand. If you tell a patient the side effect beforehand, you're like a wizard. But if you don't tell them, you're the bad guy to both your staff who's fielding all of the triage questions, but also to the patient that says, why didn't anyone tell me about this? Latanoprost, that is in a preservative-free format. IUZA is on the market. This was FDA approved at the end of 2022. This was the first preservative-free latanoprost. I put an asterisk next to it because it's not the first preservative-free prostaglandin analog. Travoprost is out there under the trade name Xioptan, and it is preservative-free, and it comes in a vial, just like this comes in a vial. Both of those medications are not covered by insurance, and it's available through specialty pharmacies. For some of my patients have true BAK allergies, or those that have had corneal transplants, or they are a little extra sensitive, they have responded really well to this, and they seem to like the ability to have this delivered straight to their home. So that is the other one. This medication here, I watched it go through the phase 2 trials, the phase 3 trials. I watched the new drug authorization get filed, the PDUFA date, and then it finally got approved at the end of 2022. And then I still haven't seen a sample show up in my office. So I am waiting with bated breath for Omlanti is the trade name, but it's Omnipegal isopropyl. And what this is this is a non-PGA selective EP2 agonist. What this means is that this drug targets the conventional and the uveal scleral outflow, but also it is able to do so in a fashion that does not have the side effects of prostaglandin analogs. The picture that you're seeing here on the right is showing you what some of the effects of long-term prostaglandin analog use look like. This is a glaucoma patient that had a trabeculectomy in his left eye and has been using a prostaglandin in the right eye to keep the pressure controlled. And if you look, you can actually see this recession of the lid, this periorbital fat atrophy, and look at the difference here. This is the natural fat pad of this patient's eye on his left eye. And if you look over here to the right, you can see how receded that is from his long-term use of the prostaglandin analog. You can also see the lid here over the top of, almost in contact with his lash line here. But look at how that lid is kind of tucked under and receded because of the periorbital fat atrophy. What I also want you to look at is the coloration of the eyelids. You can see here this dark kind of purplish periorbital hyperpigmentation. These are all, and you can also look at the length of the lashes here on the right side of the eye versus the left side of the eye that had the trap. So some of the side effects, one side effect, two side effects, may not seem very, much of a difference, but for some patients long term, it can actually change how they look as a person. And that can change, impact their identity. They're already dealing with a blinding disease, but to realize that they're on a medication that's also changing the way they look can also impact the way that they feel and can impact their mental health. So this is a neat molecule because it was not associated with this periorbitopathy. And it's been in use in Japan since 2018 under the trade name Ibellis. The projected availability, I'm still waiting. So once I know, then I'll let y'all know. We're going to talk about some investigative molecules, just so you kind of have an idea of what's out there in the clinical pipeline. This is a drug that is no longer in the pipeline, but I was very excited for this drug. If this was FDA approved, it would have been the first once dosed alpha adrenergic agonist, our purple tops. The new drug authorization was filed at the end of 2022, and this medication was dosed once daily versus Alpha-GAN P dosed 3 times daily, and it achieved functional equivalency across all time points. So it was exactly the same. So a 1 dose a day versus a 3 time a day for this medication. And even though the FDA didn't have any issues with the clinical safety or efficacy, they said all the data looks fine. The CRL that they issued had problems with the active pharmaceutical ingredient manufacturing facility. So it actually had to do with the manufacturing of the drug. And ultimately, the FDA decided not to approve this drug. So this just goes to show that a company could put literal billions of dollars into research and development to have a drug that all of a sudden gets to the end of the line. Everything can look great, but because of the way that it's manufactured, the FDA will say no because its job is to keep the American public safe. So that would have been awesome for patients that are on TID dosing of an alpha adrenergic agonist. This is a medication called bimatoprost Grenade. Remember, we had latanoprost Bunod. This is bimatoprost Grenade, and what this is a nitric oxide donating bimatoprost. The Dolomites and the Mont Blanc studies were looking at the concentration. Remember, phase 2 is looking at concentration. And the Dolomite study showed there was non-inferiority and also superiority to latanoprost alone. I was involved in the clinical trials for this particular molecule at my practice, and so it was very exciting to see something that this was the first time that head-to-head against latanoprost. Now latanoprost is the new gold standard, that glaucoma medications versus timolol used to be what the FDA deemed that the new molecules had to go against. The Mont Blanc study, these are highly powered studies. You can look at the N value. You want to see a high number N that lets lots of patients involved in the study, and this achieved superiority at 4 of the 6 time points that the investigators were looking at, but there was greater numerical reduction at all of the time points. Recently, there was the Whistler 3B data set. This came out just about 2 months ago, and it was investigating the aqueous humor parameters that were stimulated by nitric oxide. And the investigators found that they were statistically significant to make sure that it wasn't the concentration of the bimatoprost that was causing this change, but actually nitric oxide in and of itself. And by change, I mean the lowering of the IOP. This study was also looking to investigate whether nitric oxide could potentially impact the episcleral venous pressure, and there was no statistically significant change in the EVP. But we do know that that nitric oxide metabolites and those parameters were present there within the aqueous humor, and this is likely responsible for the increased IOP lowering effect that was seen in the previous trials. The Denali study was the one that I was involved in, and that's a phase 3 study. The top line data is expected soon. And what's interesting to know is that COA gained exclusive rights to develop this drug and to distribute it in the United States and other territories. So I think that everyone should kind of be on the lookout for that molecule to be out, because it may be yet another tool within our toolbox. Stewart Therapeutics has a ST113 molecule, and this is very interesting. This is a synthesized collagen mimetic peptide. This molecule was shown to have true neuroprotective, but also neuronal repair capability in animal models. And those animal models were simulated with both POAG and normal tension glaucoma. This is super exciting because I remember when I was a student, there was this talk of, bromonidine being a neuroprotective agent. And the papers did not pan out to show that there was true neuroprotective capability of that molecule. Here's a molecule that may not only protect the optic nerve, but may have the ability to repair damaged nerve. And right now, this molecule is an investigational new drug enabling stage. And so I would keep your eyes on this, because I watch the space all the time. As a former analyst, it never leaves you. And this space of glaucoma therapy is very exciting, because the optic nerve is one of the largest nerves in the body. It's a direct pathway, not only from, the eye into the lateral genicula of the nucleus, but it is a very long axon, one long continuous axon. And if a molecule has the capability to repair this damaged axon, which can be evaluated quantitatively and qualitatively for the eye, it has the potential to repair the neuronal damage in other parts of the body, including the brain. And so I watched the neurological literature in addition to the ophthalmological literature. This is a molecule to keep your eyes on. This is a honorable mention, I'll put it. SB100 ophthalmic emulsion was a drug that was investigated by a company called Sky Bioscience. It is a cannabinoid receptor. And we have cannabinoid receptors throughout the body, and there are cannabinoid receptors in the eye. And their phase 2A investigator, remember, phase 2 was looking at concentrations of the drug, and it was looking at 1% versus 0.5 concentration. And unfortunately, this drug failed to meet the primary endpoint of lowering the pressure in patients that had both POAG or ocular hypertension. Even though this, and then Sky Bioscience stopped this pipeline and did not investigate further. This is the farthest along that a cannabinoid receptor has made it within the FDA clinical trial process or the phase data sets. And so we're out of human safety models now and into actual looking at concentrations of molecules. So I think there are companies that are investigating to look into this space to utilize any available receptor that's out there, including the cannabinoid receptor. So like I said, I give that molecule an honorable mention. And finally, the PER-001. This is a very interesting drug/vehicle that is being investigated by a company called Perfuse Therapeutics. If approved and making it through phase 3, it would be the first in class endothelian agonist, and it's an intravitreal implant. The phase 1 and 2A data investigated, remember, human safety, and then also looking at dosage. It was looking at a low and a high dose of the drug, 33 patients. It was a single intravitreal implant that was followed through for 24 weeks. Not only did every single participant have increased ocular blood flow, but they also had improved visual function and anatomic structure. investigated and analyzed with ERG. And so this was safe and well tolerated. There were no safety issues with this. This drug is looking to be investigated for glaucoma and diabetic retinopathy dual indication. So I have plenty of patients that have glaucoma that also have diabetic retinopathy, and my retina surgeons have plenty of retinal patients that have glaucoma. And this would be a drug that could simultaneously treat both. And the papers were just published within the last few weeks of this top line data for the phase 1 and 2A. This is very, very exciting and may allow us as optometrists to be able to continue co-managing patients like we do with glaucoma and with retina, and to be able to have something that's working 24 hours a day in the patient's eye, utilizing a very unique mechanism of action. So we talked about some therapies and some drugs that are on the market out. We talked about some investigational drugs. The 2 poll questions coming up, we'll talk about drugs that we already have in our toolbox. So the first poll: which of the following drugs utilizes nitric oxide, which is an endogenous molecule found in all major human organ systems? And I'll give you guys time to take a look at the question. And then if Joe has fielded any questions that may have popped up while we
have not
look at our discussion.
We have not.
Oh, good. I hope that I have everyone is not asleep, but if you are, at least you have some, you can absorb through osmosis, which sometimes in optometry school, when I would fall asleep on top of my study materials, I hoped would happen.
So the polling question is open. As a reminder, this is a synchronous virtual course. So there's the chat box right above the polling questionnaire. You can put your questions in there. For Nora Lee, myself, Joe, well, you can all tag team these tonight if needed. Obviously, our speaker tonight would be the first to answer the question. Polls or questions are rolling in here nicely, or the responses are rolling in nicely here. So I will give it another. I'll give it another second or two. I really like the way you targeted the trabecular meshwork. You know, glaucoma is a disease, at least high pressure glaucoma is a disease of outflow. The trabecular meshwork, you know, even though the nerve is dying, that's why we see all this MIGS and kind of emphasis on that trabecular meshwork, nitric oxide. You're going to be talking about MIGS, I believe, so I'm not going to let the cat out of the bag there, but
it's the hot space, Greg. It's where the party's at. The party is in the trabecular meshwork. That's where the hot spot is.
And then, you know, that was San Diego Academy. I was helping launch the the new glaucoma, the EP2 receptor. I was geeking out on that, and it's
you and me both.
And I was like, oh my gosh! And I forget his name, Michael something. He did his PhD in trabecular meshwork, and he taught. I mean, I sat down for 2 hours. I learned so much about the trabecular meshwork and receptors, and then the medication got approved and never made it. I don't know what happened, but all right. So here we are. Your answer was 62% with Vyzulta. Rhopressa was 28%. And then down the line is 3% and 7%. So the winner was Vyzulta at 61%.
And that is the correct answer. Am I allowed to say that if it's the correct answer?
Oh yeah, absolutely. Okay. This is a learning. Okay,
experience. Great. All right. Poll question number 2 coming up.
Okay.
Which of the following drugs is a Rho kinase inhibitor that increases the aqueous outflow via the trabecular meshwork, also known as the traditional pathway? So the Rho kinase inhibitor.
And that polling question is open.
And for people who are putting their answers in the chat, that's not the right place. Go below the chat, the icon for polls. That's where you'll see it. We don't respond to the polls in the chat.
Looks like you got a clear winner here. You've got maybe the Rho kinase and the name with Rhopressa. We've got 97%.
Awesome.
Going with Rhopressa. We have 0 for Vyzulta and 3% for Iuza.
Excellent. Great job, guys. All right. What about lasers? I talked about all these molecules, but lasers are the hot topic. I recommend lasers all day, every day in my clinic. All I do is medical glaucoma, neural eye disease. And I always have to say, I'm an OD, not an MD in the state of Florida. I cannot shoot a laser, but I could in 15 other states. So congratulations to all those states that are experiencing scope expansion for your ability to utilize lasers. Now, why are lasers important? The laser in glaucoma and ocular hypertension trial was a trial that investigated ocular hypertension in POAG patients to see if there was a difference whether patients that were treatment naive, if they got SLT or laser first, selective laser, trabeculoplasty, or if they were assigned drops first, what would happen to these patients, and was there really a difference? Up to this point, Europe had been using laser first. It was laser first, then drops. And in the United States, we were drops, 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 laser, maybe some more drops, and then surgery. Laser was being saved till the end. And so this was a really nice study. It was published in 2019, and it looked at cost, efficacy, and safety. But this is actually one of the first studies that looked at quality of life. And what it found was that patients that received the laser first, they had disease stability, but they had a greatly reduced cost burden. The amount of money that was coming out of their pockets, whether that's in buying drops, going to the pharmacy, follow-up appointments to make sure that they were using their drops. And so there's a lot of burden that we're adding into these patients. It might seem easy just to say, add on a drop, add another drop, add another drop. But for each of those drops that you're adding on, it's like putting a brick inside of a patient's backpack, and you don't know what else that those patients are dealing with in their lives. So our goal as doctors is to limit the burden that we're putting on the patient and to not do any harm. And so the ability to recommend a laser first with backing from a nice publication like this has really been awesome. The AAO, the American Academy of Ophthalmology, and the EGS, they all now recommend SLT as first-line treatment for primary open-angle glaucoma, and I do as well in my practice when patients are first diagnosed. And the laser itself, the SLT laser, used to be a laser where a patient had to sit tight in a microscope. You had to aim the light into the TM. The doc has a little foot pedal, and they're aiming in the light and shooting the laser into the TM while the patient holds still. There is a better way to do things now. There's something called the DSLT, the direct selective laser trabeculoplasty. My practice in Florida was the first in the state of Florida to acquire this laser. I watched it. It was actually created in Israel and was called a Belkin laser, and a company called Alcon here in the United States acquired it, and it's distributed in the United States now under the name as the Voyager laser. So if you hear DSLT in the United States, it's actually the Voyager laser that Alcon has. And this clearance was granted in 2023. This is a Q switch 532 frequency double Nd:YAG, and the energy is directed through the sclera to the TM. No gonioscopy lens required. So if you look here in this little picture here in the bottom left corner, the old SLT, you had to put the gonio lens on the eye, and you had to have the patient hold still, and you shine the laser, and you actually have to be able to visualize the actual TM. Now, if a patient had no pigment in the TM, sometimes it would be hard to find it. If a patient had an iris that was being bowed a little bit forward by the physiological lens, a lot of things could impact the way in which the power is being aimed. Now the laser treats through the outside of the eye on the sclera, and it treats in 3 seconds. This is something that has really been a game changer for us in my practice. I talked to my cataract surgeon is actually who got this for us. So because I explained to him, I really advocated for it. And it really just goes to show when you have a good relationship between optometry and ophthalmology, it can really make a big difference in the care that you're able to provide for your patients. So even though I can't shoot this laser in Florida, I've got a surgeon that's a cataract surgeon that's willing to shoot this laser for me, as for as many patients as needs to. And the patient doesn't have to sit down. They walk right up to the laser. There's a speculum that's put in place, and doc hits the button, and within 1, 2, 3, boom, patient is treated. It sounds very cool, but is it the same as SLT? The study called the Gloria study investigated both the SLT versus DSLT, and it found that both were equally efficacious and they had a similar safety profile. So whether you're aiming the light through the TM in through a gonio lens, or you're treating outside on the sclera at the limbal margin, the lasers work the same. One's not better than the other, one's not inferior than the other, they're equal. One caveat that I will have is that we have found that patients that have arcus at their limbal margin and patients that have large pinguiculas or pterygiums, sometimes the SureTrack algorithm that's using those conjunctival vessels has a hard time focusing. So I actually defer to traditional SLT for those. But my patients that have had traditional SLT versus the DSLT, they really do like the DSLT. This is something that is in investigation in the United States. And the question is, would optometrists that are in laser states be able to use this? We'll see. This is called FLIGHT. This is the femtosecond laser guided high precision trabeculotomy. And what you're seeing in that bottom left corner is a real-time image that should not real time now because we're giving this lecture, but what a surgeon would see. And you actually can see the light in the TM. Scleral spur, Schwalbe's line, and this laser is creating customizable non-incisional TM channels. You get high definition gonioscopy, but you're getting real-time OCT of the anterior chamber, and it's currently under FDA clinical investigation. So this is a little bit more. If you look at the brown area and think of that as being the trabecular meshwork, the femtosecond laser is aiming in, and it's actually creating this perfect pocket that goes straight into Schlemm's canal. And you can put them at multiple places through the TM. And on the left side, you can actually visualize this with a gonio lens and actually see these nice little perfect little pockets. The first in human safety trials, remember, that would fall under phase 1, were done in 18 eyes. These patients had a range of glaucoma. What's interesting is that at 24 months, not only did the IOP go down by 34%, these are patients that were not on any other medication, this is the procedure that they had, but 100% of these trabeculotomies, they were unobstructed. You could see them on gonio. There were no PAS or iris incarceration. So when I first heard of this technology a few years ago, I thought, wow, we're blasting little perfect spaces into the TM, but are they going to scar over? And they didn't. There were no reports of hyphema, which is something else that I also kind of expected, but that's not what happened. No pressure spikes, no corneal edema, hypotony, or other device-related adverse events. So this has showing very nice promise, and will be in more trials underway in the United States to have clearance. But if optometrists are able to utilize an SLT, LPI, those lasers, they may be able to have this in their toolbox to create these precision trabeculotomies. This is a femtosecond laser. This is different than the Nd:YAG laser that is used for the SLT. So poll question number 3. The Gloria study compared the direct selective laser trabeculoplasty to the traditional SLT and found that, was it inferior, superior, was it equal, or was there not enough data to draw a conclusion?
Okay, polling question is open.
We have no questions in the chat.
No questions in the chat.
No. Nope. Well, one question came in.
Yes.
I think people got shamed.
No, I don't want them to feel shamed. I want them to feel excited.
They feel like they should respond. It's good. It's a very good question. If a patient has had SLT, can they have DSLT later?
Yes, they can. And what's really
Good
nice
question.
Well, yeah, this is an excellent question, because remember, the mechanism of SLT, I explain it to my patients like this. It's like if you have a storm drain at the end of a sidewalk and you shine a flashlight down the storm drain. I explain it's a cold laser. It's not burning anything, different than ALT, which was a predecessor. We're shining a flashlight, and I kind of explain it. We're using the bat signal to call the immune system to the area. They get in there and they scrub out the drain like scrubbing bubbles, and they go down the drain. And so that effect doesn't change or doesn't damage the tissue. It doesn't scar the tissue. And so the mechanism of action, whether you apply the laser directly in through the limbal margin or you're shining it straight into the TM with the traditional SLT, is the same mechanism of action. So yes, if a patient has had traditional SLT, they can have DSLT, but remember, they're using the same MOA. So if you do the procedures back to back, you don't get an additive effect. But I've had patients where the SLT has worn off I tell patients your SLT is going to last about 2 to 4 years, maybe up to 7 years or more. But once it wears off, we can always repeat with the laser. So a lot of my patients, I see the pressure go up. It's not that the glaucoma is going out of control, it's just because their laser is worn off from a few years ago. So then I have them do the DSLT and they get about the same pressure lowering effect. Great question.
Joe, any other questions?
We're good.
Okay, let me display. We have 3% going DSLT was inferior, and then B was 21%. The winner at 71% was C, where DSLT was equally efficacious to SLT. And we have 5% going not enough data to draw a definitive conclusion. So might want to explain that one.
Excellent. They work equally well. The DSLT and the SLT were equally efficacious. One wasn't better than the other, or one wasn't inferior to the other. They actually work the same. So at our office, we've got one office location that has the regular SLT, one that has the DSLT. And for optometrists, if you're in a laser state and you want to be able to offer this technology to your patients, you have the ability. We have it on, we don't even have it in the ASC. It's downstairs in an exam room. And it's a really, really nice technology. And so for optometrists to be able to offer SLT as first line, the light study shows that those patients are going to have a better quality of life and a decreased cost burden in time. Let's talk about one of my favorite subjects, which is MIGS. MIGS can seem super scary, kind of like when I think about all of the drugs that are out there in the retina world right now. I get very overwhelmed, but MIGS is actually a very, very cool topic, and we're going to dive a little more into it. So just like I did with the therapeutic timeline, I made us a surgical timeline so we could see how things have evolved in the surgical landscape. And on the far left, we have traditional trabeculectomy. That's like a trab. When you hear surgeons talk about a trab or the bleb, and the glaucoma drainage devices like the Med glaucoma valve and the Multino valves, those are all kind of in the 60s, 70s, and the newer technologies, newer iterations came along. The ALT was an argon laser. It was a hot laser that kind of burned holes in the TM. The SLT didn't come along until 2001. Think of it like the cold laser that kind of treats the drainage of the eye to scrub out the garbage in the TM. And then in 2012, after I'd already, as I was graduating optometry school, trabecular stenting came along. We slowly started realizing that the TM is the ultimate place of dysfunction for the disease. So targeting it through various methods, like we were using with the SLT, was the way to, provide additional avenues of outflow. So a company called Glaukos made what's called the eye stent. They really kind of kicked in the door for these new devices and kind of started what I call the MIGS movement. In 2016, there was a term called minimally invasive glaucoma surgery. And what that was a controlled trabeculectomy. So like the trabs of the former years could have different, different appearances of blebs, different longevities. Some patients heal differently. But what this did was allowed a surgeon to have a more controlled trab. And that is a MIG called a Zen. And then in 2020 onward, we had these sustained release. So they're under the MIGS umbrella, but they're not outflow devices. These are ways in which we put drugs in the eye instead of on the eye. So MIGS is a kind of an umbrella. And some of the key features of MIGS, I made this acronym ASSET because they are an asset to us that manage glaucoma. Most MIGS use an ab interno approach, meaning they're going from the inside out when they're placed. They are safe. There's a swift recovery. They're efficacious, and there's minimal tissue disruption. So all MIGS that are available on the market are assets to us as we care for these patients. These can be performed with cataract surgery or as standalone, depending on what the FDA has cleared them for. They target all aspects of aqueous production and outflow. So I know we focused on the TM, but there are MIGS that are out in the investigational pipeline that are utilizing other avenues of getting the fluid out of the eye. And they can mitigate therapeutic noncompliance for a variety of reasons. This paper was published this year, and Christine Funke and her colleagues, this is an interventional glaucoma consensus treatment protocol. This is one of the first times in my 10-year clinical career that I've seen ophthalmology push, champion a paper about kind of the recipe book on how to manage glaucoma. And what I find very interesting is that, drops were discussed as being, always there, or being a bridge between MIGS, and SLT as the first line. And I find it very interesting as someone that lives and breathes optometry. I love our profession. I'm actively involved in our AOA, that ophthalmology is championing SLT as first line, so that we can care for all these glaucoma patients. But there are facets of their profession that are very quick to push away and push against optometry having SLT in our therapeutic arsenal. And so you can't have it both ways. You can't want SLT to be there for all these patients because there simply aren't enough ophthalmologists to care for all of these glaucoma patients, especially glaucoma surgeons, but also want to keep SLT out of the hands of optometry. And that's just my 2 cents of advocacy on behalf of the profession. And I say that in MD spaces all the time. I'm very outspoken about how capable I think that we are as a profession. And so this treatment protocol really did look at SLT first and MIGS along the journey, with drugs being and topical therapies being the bridge. I think this is the right step of the right direction of the mindset. But like I said, you have to be able to, I don't see any optometrist on the authors of this particular paper. And I think that you really do, in order for us to best care for our communities, we have to continue working together. So let's talk about some sustained release. Remember, there was a little category at the end of that timeline that were drugs in the eye versus on the eye. So travoprost sustained release, it goes by the trade name iDose, made by a company called Glaukos. It was approved in December of 2023. It's a little 3-part titanium implant that actually goes right into the trabecular meshwork. Think of it as like being a Glade plug-in, and you push it into the wall, and the little prong goes in there and it seats into the scleral wall. The phase 2b data showed that the average IOP lowering effect was about 32%, and this has proprietary travoprost in that slowly eludes over the course of 3 years. And so patients were given the iDose, which was the travoprost, and also the timolol arm, because remember, at this time, the FDA required companies to go head-to-head with timolol. And in theory, it provides 3 years of sustained IOP reduction. There were no serious adverse events. The picture that you see on the bottom left here, this is 1.8 by 0.5 millimeters. It is slightly noticeable for patients, but for a lot of patients that have been drop free and they enjoy staying drop free, this may be a nice option, for them. There was a study that looked at what happens when this was initially discussed. I thought you were going to be able to go change the cartridge out, just like you would change the Glade plug-in at your house and change the cartridge, but it doesn't allow for that at this point in time. The actual implant does have to come out. But the good thing is that there was an investigation that looked at the safety profile of removing this out of the TM and from being seated in the scleral wall, and there were no serious complications when that was removed. So that's a piece of hardware that's installed in the eye that slowly eludes travoprost. The matoprost TR, or extended release, sustained release, that should be an S versus a T, goes by the trade name Durista, made by a company called AbbVie, and it was approved in 2020. A lot of optometry didn't hear about it because we had the whole world collapse with the global pandemic. So this company, instead of marketing this and putting it on TV and putting it in brochures, they really focused on pivoting to donate a lot of therapeutic drugs, waive copays, and also get a lot of artificial tears for first responders. So what this is, it is built on the Ozurdex platform. So Ozurdex was a bioinert polymer matrix that's put in the posterior chamber with a steroid in it. The company thought, what if we put glaucoma medicine and put it in the anterior chamber? And it worked great. It slowly dissolves. Under the scanning electron micrograph, it looks like a little block of Swiss cheese, and it just slowly dissolves over time. And when you look at it in the gonioscopy lens, I took that picture with my iPhone with the gonio prism, and you can see it sitting in there, that little tiny, it looks like just a little tiny little cylinder, and it just sits right there into the angle. It can be performed in the ASC or exam lane, and 28 gauge injector bevel up is used at the limbal margin to place this in. The Artemis was the phase 3 clinical trial for this particular MIGS, and patients got about 32 to 33% pressure lowering, which is right on par with what the Travoprost extended release did. And this makes sense because this is a prostaglandin. It's a 1-for-1 swap for patients that are on prostaglandin analogs. There's a new paper that was just published 2 months ago called the Argos data. And in that paper, patients were given one bimatoprost extended release and were followed into the future. And at 18 months, 77% of patients were drop free. The medication is gone after 3 months, 4 months. You can do an AC tap at 5 months and there's not a molecule of bimatoprost inside of the eye. But clinically, I saw that the patients were getting really good pressure lowering effect. And we've done over 500 of them in my practice. And I tell patients, you're going to get about a year and a half out of this. You might get up to 2 to 3 years out of it. And that's actually what the Argos data was showing. So this is another reminder that clinical data that's done after something has been approved, kind of like how Connie and I did our retrospective chart review and looked into the IRIS registry data, looking at something after it's already been approved to see how it works, to get a data set. This actually confirmed what I was seeing in clinical practice about how long this worked. So there's actually a very unique mechanism of action that may involve some remodeling at the trabecular meshwork. And we'll get to that when we get to the genetic section, but there's a little teaser for why this might work longer than there's actual medication.
Before you go on, the question came in. It's a very good question.
Yes.
Any preference, Durysta versus iDose? Do you specify one over the other when referring a patient, or is that going to be up to the glaucoma surgeon?
I think it's going to be left up to the glaucoma surgeon, but for me, my initial reach is for the Durysta. Because of the molecular remodeling that's likely occurring within the trabecular meshwork. It is like an Alka-Seltzer that slowly dissolves over time, leaving nothing behind, versus a piece of hardware that's installed. For the hardware to get installed that's not done at the biomicroscope, the patient actually has to go to the OR. And the majority of glaucoma surgeons and even cataract surgeons that I know that do the Durysta do them at the microscope. So for a patient, there's a decreased cost burden. They just have their office copay for the procedure that they're having done versus the ASC fee of going in and having actual surgery. Patients that have had Durysta that have waned are excellent candidates for the iDose TR as the next step because they're already used to being drop free. So I usually like to order it in that way because of the mechanism of action. That's a great question.
Mm-hmm. I just want to feed off of your Durysta after talking to the medical science liaison, and just kind of feed off a little bit of your interventional glaucoma. You know, interventional glaucoma, for the audience out there, is they're trying to get away from drops. The drops create toxicity. The drops create damage to the front surface and ocular surface type of condition. So they're trying to migrate and get away from using drops and using SLT and using these MIGS, as Nora Lee is so well explaining. But in talking to the Durysta rep, and just kind of feeding off of the interventional glaucoma, and you got to put, bimatoprost or Lumigan, on the eye every single day, and maybe it can last up to 36 hours to show you how robust that cornea is and conjunctiva and how hard it is to get medication in the eye. Durysta is 1 drop of 0.3 of Lumigan that slowly dissolves, like Nora Lee has mentioned, over that 5-month period, but then you get the lasting effect. So just think about it. It's 1 drop of the 0.3 that's put in there, and then you get about a 1.5 year outcome from it. Pretty amazing, as opposed to putting a drop on the eye every single day.
It's really remarkable. And what's interesting is that what was found in the Durysta clinical trials actually spurned the creation of this company called Spyglass Pharma. And this MIG called, I put under the MIGS umbrella, but it is a IOL with bimatoprost eluting wings. The wings are mounted into the sides of the IOL, and the dose, later on, we'll look in some of the genetic section, but the ability to remodel the TM was dose dependent. So actually, some of the researchers that were involved in the clinical trials for Durysta work for this company now to create this bimatoprost-eluting wings in this IOL. And the first in human feasibility study, all the patients achieved a best corrected distance visual acuity of 20/30 after their cataract surgery. They had a 45% IOP reduction from baseline at 9 months. So these patients, remember, Durysta was a 1-for-1 swap, about 32 to 33% IOP lowering with the Durysta. But with a higher concentration of bimatoprost that's eluting over the course of 3 years, patients got up to a 45% reduction. Now, is some of that IOP reduction from taking the lens out? Of course it is. But 100% of these patients were off of all IOP lowering drops at the 18-month mark, and the study is still ongoing. And so this, we were involved in clinical trials for this as well. But I am very interested to see if the IOP lowering effect lasts beyond those 3 years. And in talking to some of the researchers that are involved, with creating this, that's the idea, is remodeling the TM. And it's a very, very beautiful design. And I think that we're going to see more of this where we put the molecule in the eye versus on the eye.
In that picture, is the medication in the blue part of that?
Yeah, the little the little blue pads at the end of the haptics is kind of on the edge of those wings. And then in the bottom picture here, you can actually see those little wings here as they would be kind of at the base where the haptic joins the body of the IOL.
Perfect. Thank you.
Very, very cool thing to kind of be on the lookout for. Let's talk about MIGS. So MIGS can be very scary and very confusing, and it does not have to be. If you think about MIGS as being, those assets that we have to help our patients, if we think about where the MIG is working, in what space it's working anatomically, it helps you mentally kind of keep them in different buckets or different categories. The first category is the trabecular outflow, where we know the ultimate dysfunction is of the disease. And we have the iStent Infinite and the Hydrus Microstent. These are the two that are on the market right now that are actual hardware pieces that, as an optometrist, you can actually see when you're looking into the eye. Canaloplasty and goniotomy falls under trabecular outflow, but each of these categories has about a half a dozen different devices made by different companies that either, it's almost like taking a little drain snake through the TM or Schlemm's canal behind the TM and cleaning out the trash, or tearing out all of the bars of the drain at the end of a sidewalk. And so there are different devices that accomplish that, but I'm going to focus on the things that we can see as optometrists, because I never want one of our colleagues to look into an anterior chamber and not know what they're looking at. If you do that, then docs like me and Justin and Mitch, we fail because we haven't shared that information to make sure that we've educated our colleagues on what you're seeing inside the AC. The subconjunctival mechanism of action of getting the fluid out above the conj, that's where the traditional trab used to work, and the Zen gel stent is that controlled trab. The PreserFlow is still an investigation, and everything else with the little asterisk next to it is still investigational. So we have the PreserFlow that's used in Europe, but it is still investigational. The supraciliary space is a place that is ripe for harnessing the power of that space, but every device that's out there are still in investigational for the United States. So let's talk about the stuff we can see. So utilizing the trabecular outflow, the iStent Infinite is the newest iteration of the iStent. Remember, the original iStent came out in 2012. This is like the iPhone. You get better and better versions of it as it goes along. You have this stent that's kind of flanged at the bottom. It looks a little bit like a golf tee with the flange on the end of it. And the iStent Infinite is a trio of these stents. So we started out with one stent. I remember around 2015 and 16 at the AGS meeting seeing a paper where one of the glaucoma surgeons was doing 2 stents and saw that it worked just as good and even better. And then now we have the possibility for 3 stents to span across 6 to 8 clock hours within the patient's angle. Average IOP reduction about 7 millimeters of mercury, and this can be performed at the same time as cataract surgery or as a standalone procedure. This is really important because you may have patients that come to you, anybody that had an iStent had already had cataract surgery, so they were already pseudophakic. in your optometric practices, you may have patients that come in that may have stents in place that aren't, that may still be phakic, that aren't pseudophakic, because this can be done as a standalone. This is why it's so important to use your gonioscopy prism to look into the angle, because if you don't look, you won't see. I teach my clinical externs from optometry schools, and even the residents and fellows that shadow. You look for the shiny, and this will be very, very easy to see. You'll see this nice bright silver shine shining back at you. The post-op period for the iStent Infinite is 90 days, and there's no major deviation from a post-op cataract surgery schedule. Patients stay on their drops the same ways. You want to be able to document where you see the stents, and if they're obstructed, very unlikely for them to be obstructed. They'll look like a little shiny silver donut when you look at them. And there's very nominal complications. This has been something that this company has fine-tuned over the course of a decade. And we have all of my cataract surgeons are doing this iStents. The other stent that you might see in a patient's eye is called the Hydrus microstent, and that is managed by a company called Alcon. The FDA approval was in August of 2018, and this is an 8mm trimodal nitinol stent, and this is pretty large. 8mm is a large space or a large device to go into the angle. Oftentimes you will not see it unless you look into the as you're looking into the TM. Here you can see it shining back at you, and you're just going to look for the opening here, this inlet. And these little windows, the scaffolding, it actually mounts and goes straight in through the TM and mounts into the scleral wall, but the inlet is what you'll look for. So as you move and look through the TM, you'll find the inlet, and then just follow it along, and you'll see 1 window, 2 window, 3 windows, and you'll actually see it sitting there. So it's really, really beautiful when it's put into place. And it's one of the favorites of one of my cataract surgeons. The post-op period, this is indicated for mild to moderate primary open angle glaucoma. That's really important because a lot of these devices are not approved for severe glaucoma. The same thing with if you have a patient that's severe glaucoma and you're billing and coding, CMS is going to be a lot more lenient on you doing the amount of RNFLs and the visual fields on moderate patients than they are severe patients, unfortunately. So always kind of keep in mind, as you're coding. So don't be alarmed. Maybe you have a patient that you feel might be severe. They come back with a moderate indication from their glaucoma surgeon, and it doesn't change anything about the way that their glaucoma is. But in some instances, sadly, for insurance to cover, the patient has to be coded as a moderate patient. Post-op period is 90 days. There is no combo drop. We do a separate steroid if this is, and so that we can taper and adjust for inflammation. Some patients will get an inflammatory response, and I watch for that. And then I slowly down titrate the topical glaucoma medications afterwards. The documentation, you want to look at what quadrant it is. And once you have a surgeon that's doing this, you'll know what quadrant he or she always wants to put it in, and you can look for it. Some patients will get a high FEMA on post-op day 1. That's not uncommon, but it doesn't impact the outcome at all. That steroid will be able to calm, and you have like almost a nice open place for all of that high FEMA to be able to be carried out through the trabecular meshwork. Moving into the subconjunctival space. So we were looking in the TM. Now we went low. Now we're going to go high, up above the TM into the subconj space. The Zen microstent was the first MIGS, and this is a minimally invasive bleb surgery. This is a hydrophilic gelatin stent, and it's making a perfectly controlled bleb here. So this is going in, our TM is way down here, but this is going above and overshooting right above the conj. This patient had up to almost 9.2, millimeters of mercury at post-op month 12 at about a year. The indication for this is POAG. This is one of the only MIGS that actually has an indication for pseudoexfoliative and pigmentary glaucoma. Allergan, or the company AbbVie, had enough pigmentary and pseudoexfoliative patients to get this on label, which is great. It preserves the conjunctival real estate for the future if a surgeon needs to do a tube or a shunt. And prior to the approval, this was already used in all across the EU and in Turkey. So if you don't look for this, you might miss it. So this is a picture I took in the microscope of my patient here. And if you weren't looking, you might not see this. This is different than the big giant blebs that you might see before from a traditional trabeculectomy surgery. I tell my clinical externs, if Barbie was drinking a boba tea, this is what the straw would be in the boba tea. And that's about what it looks like. It is very different than the big tubes that you see coming from the Bearville and the Ahmed valves. It's going to have a flat end here. The Bearville and the Ahmed valves are clipped in surgery so that they can have a bevel that's up. This is going to be flat, and it should not touch the cornea, and it should not touch the anterior face of the iris. But you'll want to look for that. There'll usually be superior temporal superior nasal. Here's another one here. And if you weren't looking for it, it kind of blends in, with the patient's, anatomy, but it will shine back at you. It almost glows a little bit of a yellowish color, a little yellow tint. The post-op, these patients are on topical antibiotics 4 times a day for a week, and then a steroid taper. You want to watch the bleb appearance and look at the location of the tube. There should be no contact with the endothelium, and be on the lookout for any inflammation, wound leaks, tube occlusion, or device migration. These are very rare, but if they happen, I want you to know, if you have one documented and all of a sudden it disappears, look for it. It might be, maybe it migrated out and it's hanging out in the anterior chamber. I haven't seen that, but I have heard of it happening. It might have migrated up and in underneath of the conj and then send it back to the surgeon. There's a study called the Gold Standard Pathway Study that looked at the Zen gel microstent versus a traditional trab, and it was highly powered at 202. Patients that had the Zen had greater comfort and a better safety profile, because there's always a risk for endophthalmitis and hypotony, sometimes with patients that had the traditional trab. Patients were more comfortable because the bleb was very predictable. Some of the limitations, about 55% of these will require reneedling within a year and a half. So if you have a patient that's had one of these, be on the lookout. If the pressure goes back up, it doesn't mean it's completely failed. It's just not working as well as it was. Go ahead and you can put on an IOP lowering agent, like a life jacket, for the surgeon, and send them straight back to the surgeon, because they have a very good probability of coming back to life when they're reneedled with MMC. A similar stent.
Hey, Nora Lee.
Yes.
Just for clarification, the gel stent doesn't have a hole in it, right? It's more candle wicking, creating that bleb, or does it have a tube in it?
It is a tiny little hole in the center of it. So it actually is a little stent, just like a tiny, tiny drinking straw. So there are some devices that are wicking, which we'll get to, but there is a teeny tiny little hole that's in there, and it can become obstructed. And I've seen surgeons zap them with a YAG laser to clear out a little fibrosis, and then it opens right up. But that's an excellent question, because when you look at it, it is so small, it's hard to even visualize how any fluid could be drawn through it. But it does have a little hole that goes all the way through, like a baby drinking straw. Same thing here with this PreserFlow. This is CE marked in Europe, but it's awaiting FDA approval. This does have a little bevel on it, on the end of it, and it is just like, has a little straw. It has these little wings here, and what those wings do is those fins kind of seat it in so it can't move forward or backward. 76% of patients in their clinical trial got pressures below 17 millimeters of mercury, which is well below ages. But this is a SIB stent, and all SIBs does is it stands for the polystyrene block, isobutanol block styrene, and all that means is that's the type of material that it's made out of. And it was actually first used in coronary stents with really great success. And we're still waiting for that to be available to us in the United States. Now we're going to change gears and look into the supraciliary space, taking the fluid down. So we looked at going through the TM with the iStent and the Hydrus. We looked at going above with the Zen and with the PreserFlow. Now we're going down under into the supraciliary space. AlloFlow is CE marked in Europe. 80% of patients in the clinical trial got IOPs below 20 millimeters of mercury. And what this is, it's a 5 by 0.5 allogenic. It's microtrepined. It's a scleral allograft. So this is actually scleral tissue that has been microtrepined. You can see how teeny tiny these little fenestrations that are in there. And it's utilizing something called cilial dialysis. So we're trying to open up the space that allows for filtration into the suprachoroidal space. It uses the cyclos pen system. All that is kind of like a little fancy kind of tweezers that hold the little piece, and then it gets pushed into the ciliary suprachoroidal space. We had this available at the AGS meeting, and I watched my surgeon do this as a wet lab, and she is so great that she did this. And then she goes, okay, what's the next step? And they were like, oh, it's done. It literally took her about 2 seconds to do. And right now we're still waiting on FDA approval, but what it's creating is a cleft. So on gonioscopy, if a patient has this, you're going to see this tiny little, almost this little rectangular, it almost looks like a thin little stick of gum. And you're going to see it here. And what that's doing is it's opening up that supraciliary space like a wick, and it's allowing that fluid to be drawn through there, to be pushed out in a very controlled way. And you can use 2 at a time to make an even larger cleft. The mini-ject is the same concept. And you can see here's a better picture of it kind of going down. And this little green mark is actually on the device to show how far it needs to go in. This uses a medical grade silicone, and it's kind of got this honeycomb appearance. And what happens is that it is fenestrated like this so that the host tissue goes in and it becomes part of the host tissue and becomes incorporated. It is made considerations for the endothelium by putting that little green mark on there so it doesn't go through. This is also something we're waiting for approval. And this intercell uveal spacer called a CID, it's in investigational studies in the United States, and 81% of patients in their phase 2 trials achieved an IOP less than 18 millimeters of mercury without any medications. And so this is a little plate that's going in trying to utilize that space with a corrugated surface. So those are all kind of things that are out there just to have on your radar. But the main things that you're going to see are shiny hardwares that are in the TM, or you're going to see the tiny little Barbie drinking straw superiorly called the Zen. All the other things we talked about are not yet in our surgeon's toolbox. So we're heading toward the end, guys. We're on a marathon, and I can see the ending in sight. Poll number 4, which of the following FDA approved MIGS uses the subconj space and might require the referral back to the glaucoma surgeon for reneedling? So remember, about 55% of these need to be reneedled, meaning that the surgeon needs to put a little bit of mitomycin C to kind of bring it back to life.
And you're good on questions. We're all caught up.
Great.
The poll is open. The answers are rolling in. I think this one's taking a little bit longer than people are thinking. Thanks for participating in the polling question. This is the synchronous part. This allows us to interact, get the questions, interact with the questions in the box, interact with the polling questions. This allows us to be synchronous, live, just not in person. All right, I think we got a pretty good trend going here. I'm going to display the results. We have Durista going with 9%. We have iStent Infinite going with 27%. 20% was your Hydrus microstent. The winner at 43% was your Zen microstent.
Excellent. And when I was trying to remember this, I always thought X marks the spot, and X marks the spot where my surgeon might have to go in and reneedle and put the mitomycin C. So it's a kind of an easy way to remember it for the future. All right, we're in the home stretch, guys.
So they use mitomycin. Probably a good way to remember is they use mitomycin when they do bleb procedures, at least back in the day. And this is kind of a bleb procedure, the Zen. So this is where the mitomycin might correlate to help those to remember. Is that correct?
Yeah. Spot on, Greg. It is a bleb, and you're bringing the bleb back to life in the exact same way that you would bring the traditional trab back to life, for the bleb on a traditional trab back to life. All right, let's talk about some data. Peer-reviewed research is exploding in the glaucoma space. I mean, I always have things to read, to listen to. I keep up on all this stuff, so y'all don't have to in the audience. come to any of my lectures, I enjoy reading this. I was an analyst. My job in the military was to analyze data and to give you what's relevant. Initially, I had a bunch of papers I was going to talk about in this section, but now I actually decided I only want to focus on the papers that have clinical relevance, things that might impact you in day-to-day clinical practice. And one paper is called the Horizon Study. This is the largest long-term study of a MIGS device with continuous follow-up. So of all of those MIGS that are out there, the ones that are FDA approved, the ones that have been recalled, these out there, the Horizon Study looked at that 8mm stent, the silvery stent, to see if cataract surgery versus cataract surgery with the micro stent made a difference. Not only did it make a difference, but it made a pretty big difference. At 5 years, there were 20 to 30% improvement in the drop-free rates, meaning patients that were completely drop-free. Some patients entered the study on 4 medications, and at 5 years were still on 0 medications after the cataract and surgery was done with the microstent at the same time. The cataract surgery in the microstent patients, they were 60% less likely to require incisional glaucoma surgery, meaning they were 60% less likely to need a tube or a shunt, like an Ahmed or a Behrvelt valve. Those fail in 5 to 8 years. They have high risk of complications, side effects like diplopia, impacting the patient's psychological well-being. There's so much that goes along with those major glaucoma surgeries that placing the stent at the same time as cataract surgery made the patients less likely to require to go to that OR a second time. This is very important. The placement of the microstent at the time of cataract surgery lowered the patient's field loss by 47% versus cataract surgery alone. That's a pretty big deal. That actually showed that it's not just taking the cataract out that was making a difference, but the active filtration of moving the fluid out of the eye and keeping the IOP low and controlled 24 hours out of the day. There were no serious AEs out of 556 patients, and there was no decline in endocell count. So the Horizon data was a pretty big deal. So can data change clinical practice in long-standing surgical protocols? It can. There was a paper that was just published this year. This data was actually discussed 2 years ago at the AGS meeting. And what this looked at was the possible risk factors for acute CME after starting a PGA. The PGA warning label for all teal cap drops in the United States say that there's a risk for CME. But what was interesting is that in clinical practice, I actually didn't see this a lot. And all I do is manage glaucoma patients. So, I honestly asked even some of my retina surgeons, I said, how much CME do you actually see after PGAs? And they said, don't worry about it. Treat the glaucoma and we'll take care of it if it pops up. Like it was that much of a non-issue to my fellowship trained retina specialists that I work with. And so this was a multi-center case controlled study. 2 of my favorite places, the Kellogg Eye Institute and Bascom Palmer were a part of this trial, but almost 40,000 patients. That is a very highly powered study. And it was looking at patients that had ocular hypertension or glaucoma that were newly treated, meaning they were treatment naive and they were started on a drop for glaucoma. And if they'd had any kind of surgery, they were not included in this trial because we didn't want the CME that comes after potentially cataract surgery to be confused with the CME that might be induced by drops. So what happened? The N value is on the left side. Those are the number of patients. And actually, if you look, the number of PGA patients, because that PGAs are first line therapies. So the reason that there were so many of those patients, the percent of the number 22,000 patients, was because most patients that were newly diagnosed with glaucoma were put on prostaglandin analogs. But what was found was that the rate of CME was lowest for patients that had PGA started as first line, and they were highest for patients that were put on carbonic and anhydrase inhibitors as first line. And so when this data was shared at the AGS 2 years ago in our open, forum, this is one of my favorite meetings. One of the older doctors told the young presenter, said, you need to take this data set to the FDA because this is strong enough data that would actually change the labeling on PGAs in the United States. And this is a very powerful data set that shows that the rate of CME is actually not not highest for prostaglandins. It's actually lowest for prostaglandins for patients that are started on a therapy. This is the largest multi-center cohort study that was looking at CME with newly started topical glaucoma therapy. And so clinicians, we have to periodically reevaluate what we know. What happened at my practice, I came back from the AGS meeting and I showed my cataract surgeons this data, and they stopped doing their PGA protocol. They used to stop patients on cataract prior to cataract surgery off of PGAs and then restart them weeks later. And now we don't do that anymore because we know the risk is so low. We're on our final polling question, guys. According to the largest multi-center study that we just discussed, the incidence and risk factors of CME and glaucoma therapy, which of the following drug classes had the lowest rate of CME, that should say CME. And that means that's macular edema, cystoid macular edema.
Well, we're good on questions. Nothing else has come in.
Excellent.
We are down to our last about 4 minutes.
Great.
I think it's great to see that data. I mean, I've worked in an OD-MD place. I've seen glaucoma when Zalatan and Lumigan 0.3. We've had broken capsules. We've had, whatever complications. They were on glaucoma, and rarely have I seen CME from it. So it's great to have that data out there to kind of, take that away. I mean, we always talk about it, but it's, it's minimal, or I've never really maybe seen it once or twice.
Yeah, it's neat.
Hey, Nora, a question did pop in. Somebody asked about cluster testing with visual fields and OCT. Do you do this, something you do?
That's a great question. So I don't do cluster testing, but what I do is I always do a baseline 10-2 in addition to the baseline 24-2 on my patients. Not on the same day, obviously, but I want that baseline data. And I actually don't hesitate to do 2 24-2s within the first year of caring for the patient, because some of the data sets show that cluster testing does identify and kind of carve out some of those defects a little sharper than spacing them out over the course of years that you would be able to identify defects sooner.
Okay, so well taught. We have 76% going with prostaglandin analogs. The next runner up at 13% is beta blocker. So
prostaglandin analogs is the correct answer. They had the lowest rate of CE. I'm going to move forward through this genetic section. We may highlight this in a future discussion, but this section was discussing about glaucoma genetics. And a little bit of a review on genetics, which we don't need. You can do that on your own time, but it was just to give a little primer about our understanding of genetics and the fact that genetics are, they play a very important part in glaucoma manifestation, but the genetic architecture in adult glaucoma is highly complex. It's highly nuanced. There are factors that we are able to identify that are environmental, but we're using genome-wide association studies to look for SNPs, to be able to use polygenic risk scores to identify the highest percentage of patients that can be more susceptible to converting to glaucoma from ocular hypertension, or patients that may have the propensity to have more aggressive disease. A new study that was just published in JAMA utilized polygenic risk scores and showed that they can predict the conversion from ocular hypertension to primary open-angle glaucoma. There are papers out there that show that environmental factors can impact genetic expression. That is a term called environmental antagonistic proprietarism. The genetic expression can also predict patterns of visual field loss in different types of glaucoma. Normal tension glaucoma having central and paracentral field defects, and high tension glaucoma, patients with starting higher baseline pressures having the ability of having defects that more are concentric and generalized constriction. Site score is a first-in-class PRS for POAG. It is a test that utilized a genome-wide assessment studies to identify genetic risk factors for patient. It costs about $400, and it is not covered by insurance. Bimatoprost SR, we know, has the ability to control IOP long after the molecule is gone. And Dan Stamer's lab at Duke identified the genetic upregulation of expression of MMP1, 10, 11, and 14, and the downregulation of COL1A1 and VCAM1, which are all involved in cleaning out the trash in the trabecular meshwork. The genetic regulation actually only occurred in patients that had tissue that was derived from glaucomatous eyes, and it was dose dependent. These last 3 slides that we're going to go through look at non-traditional interventions, because I get asked about this a lot. Ginkgo biloba pops up every few years and I get asked about it. The studies that are, this particular study that is always discussed was from 2012. And bottom line is that additional investigation and stronger investigation are required. There is a hot topic of niacin and nicotinamide. I just got asked this today by a patient asking if these are appropriate to be able to utilize in glaucoma. I've seen them pop in on online Facebook groups for optometry, and I've seen some very bad advice being given. Niacin and nicotinamide, they're both forms of B3, but they're very distinct molecules. The NAME study looked at and was investigating niacin, 5,768 patients. It may have a protective effect against glaucoma, but the high dose niacin, the 3,000 international units daily, was linked to some major cardiovascular events and also drug-induced liver injury. One of my colleagues is a principal investigator for a phase 2 trial that was looking at nicotinamide. 2 patients had acute liver injury and 1 required a liver transplant. So at this point in time, the AGS and the AAO recommend against using high-dose oral niacin or nicotinamide in patients with glaucoma. Cannabis is a natural plant that has THC and cannabinoid components to it. Medicinal marijuana is legal in 40 states and recreational in 24. Smoking marijuana does lower IOP by about 25%. However, the effect dissipates in 3 to 4 hours. Patients would have to have an average dose of about 3 to 4 milligrams of THC over a 24-hour period and continual dosages in their bodies to be able to lower IOP in any clinically effective manner. So the AGS position statement is that although marijuana can lower IOP, the effects and the lack of evidence with its use alter the course of glaucoma. We do not recommend this drug for the treatment of glaucoma, but I encourage my patients to be able to utilize it for other things like depression, anxiety, whatever they're utilizing it for. The very last thing is something that's very important. Mindfulness and meditation is the only thing to date that is something that a patient can do that actually may be able to lower their IOP. These types of studies have been repeated in different patient populations and in different places within the world. And mindfulness and meditation not only lower systemic cortisol, but they increase optic nerve head perfusion and vessel density, and they lower IOP, and they actually have the ability to maybe temper diurnal fluctuation. So when a patient asks me if there's something that they can do, I actually tell them that 30 minutes of guided meditation 3 days out of the week has been shown to potentially lower their IOP, and there's no downside from a systemic standpoint. This is the last item on the list of the non-traditional. This is called the OPAP. It has achieved de novo classification from the FDA last year. It's a pair of lightweight goggles that has a tiny little pump, and it's placed over the patient's eyes, and it applies negative pressure. And it was approved for the adjunctive therapy to reduce IOP in adult patients with POAG that already had IOP of less than 21 millimeters of mercury. Patients, there were 9 in this paper, achieved a 25% reduction in IOP over the 8 hours that they were monitored. The market activities are projected for late 2025, but the problem is, just like any new technology, it might be a neat technology, but is insurance going to cover it? And if insurance doesn't cover it, a lot of times these type of tools may be out of the hands of some of our patients. I told the inventor of this, I told John, I said, send me a pair of it so I can try it. And I want to see what it's like, because I won't recommend anything to my patients that I haven't tried. And I'm still waiting for my goggles so I can give it a try. So in conclusion, I appreciate you guys hanging in with me. I know glaucoma is not necessarily the most exciting topic, but there are exciting things happening in the world of glaucoma. And throughout my career, I promise you that I will always work to make sure that you stay up to date on what's happening in the glaucoma space. We have a lot of diagnostic tools that are out there. Using them at the appropriate time and in the appropriate circumstances is important for us as doctors. New molecules are going to utilize novel pathways. Just keep them on your radar. Knowing what MIGS are out there, even though we as doctors of optometry aren't using the MIGS ourselves, we need to know what they are, what they're indicated for, so that along the patient's glaucoma journey, we can appropriately place them so that the patient can benefit from them. Our understanding of glaucoma genetics is really going to drive not only the diagnosis, but also maybe the management. There may be therapies that can alter some of the dysfunctional genetics that some of our patients have. And it's really important that we stay up to date in the glaucoma space to know what's happening in the glaucoma landscape so that we can take the very best care of the patients in our community. Are there any questions?
No, we have covered all the questions. We're good. I think we can just wrap this right up. Thank you, Nora Lee. That was tremendous. A lot of really good information in there. I think the audience really got a lot. I think I got a lot of it. I'm sure Greg did, and I'm sure the audience did. So thank you so much for doing this.
You're welcome. Thank you for having me. I really appreciate the opportunity.
So with that being said, there's a little red dot on your screen. There's chat, there's polls where you could download the course notes, and then survey right here. You can click on that and start that survey, or you can wait for the post-event email, which will go out in about 4 minutes. Again, Joe, any last questions, or are we good?
We're good.
Okay, so with that, this will wrap up the education. This was Get Up to Date in the Glaucoma Space, Technological Innovations and Shifting Paradigm. Thanks, Nora Lee, for doing this for us, doing it for Optometric Education Consultants. It was fantastic, as Joe said.
Thank you very much.